Hello everyone, do you know what's oral mucosa? Well, my team would like to talk about the oral epithelium cells of the oral mucosa. Buckle your seatbelts and get ready, because we're going through this fast. Shall we dig in? For starters, the oral mucosa is separated into several layers. The two main tissue components of the oral mucosa are the oral epithelium and an underlying connective tissue called lamina propria. Beneath the lamina propria, however, consists of the submucosa. The interface between epithelium and lamina propria is usually irregular. Outwards projections of connective tissue called connective tissue papillae interdigitated with epithelial ridges or retibags. Basal lamina is situated at the interface between epithelium and the lamina propria. The layer underlying submucosa is the periosteum and then followed by the bone. The oral epithelium constitutes the primary barrier between the oral environment and deeper tissues. It is a stratified squamous epithelium consisting of cells attached tightly to each other, thus it can be considered to consist of two functional populations. A progenitor population, which functions by dividing and provide new cells, and a maturing population, which continually differentiate, forming a protective surface layer. Progenitor cells where epithelial proliferation occurs can be split into two, stem cells and the amplifying cells. Only a small portion of progenitor cells represent as the stem cells, which the function is to produce basal cells and retain the proliferative potential of the tissue. The larger portion of the progenitor cells are the amplifying cells, where the function is to increase the number of cells available for maturation. Despite their functional differences, these proliferative cells cannot be distinguished by appearance. Maturing cells, where the epithelium mature. Cells in the basal or peribasal layers of the epithelium undergo a process of maturation as they are basically dividing and displaced towards the surface. In general, maturation in the oral cavity follows two main patterns, keratinization and non-keratinization. As for non-keratinization epithelium, there are several characteristics that you should know. The first one is melanocyte, which is responsible for the synthesis of melanin pigment. Secondly, Langerhans cells, an antigen-presenting cells and function with immune system. Merkel cells, a tactile sensory cell. Lastly, lymphocyte, which we know is associated with inflammatory response. Next, we'll be talking about the three types of oral epithelium. The main types of mature oral epithelium are the non-keratinized, as stated before, orthokeratinized, and lastly, parakeratinized. We'll be comparing between these two first. Non-keratinized epitheliums are lining mucosas, while orthokeratinized epitheliums are domesticatory mucosa. Besides that, we could compare histologically. As for keratinized epithelium, the outermost cell layer is the keratinized layer, an extremely flattened dehydrated cells where all organelles have been lost. Next layer would be the granulosum stratum, flattened cells containing keratohyalin granules associated with donofibrils. Compared to non-keratinized epithelium, the outermost cell layer are the superficial layer, a slightly flattened cells that have nuclei but less organelles, followed by the intermediate stratum, also slightly flattened cells containing many dispersed filaments and glycogens. Now, let's talk about the parakeratinized epithelium, which is also a masticatory mucosa. What's the difference between these two epitheliums? As for parakeratinized epithelium, the nuclei are retained in the whole surface layer, whereas for orthokeratinized epithelium, the nuclei and the other organelles are completely absent. That's all. Get it? Easy, right? I hope you gain a lot from this video. With that, thanks for watching.